Welcome to the Pondcast, the destination for insightful discussions and interviews on the appreciation, conservation, and husbandry of reptiles, with a focus on turtles and tortoises. Now, let's join our team of turtle nerds.
Woo! Such an inspiring intro, always gets me. Welcome to the Pondcast, episode 86. This is a big one for us here. We're very excited. We have an extremely esteemed guest with us who is known really, really well in herpetology and herpetocultural circles, but is not as well known, I think, in a lot of turtle and tortoise circles. So we're really excited to have him join us to bring some really in-depth and really impressive biological knowledge with us so that we can all learn something. So we're really excited to have Dr. Zac Loughman with us from West Liberty University. Hello, Doc.
Hi, how are you?
Can I start by asking, do you find it offensive when people call you Doc?
No.
No?
I
feel like it's perfect, right? I know you're cool and laid back. I've heard you speak countless times, and now you have a podcast of your own, and we'll get into that. But I always feel like when I call doctors Doc, it's like a term of endearment for me, but also respect, because I feel like you earn that shit. Sorry, excuse my language. You earn that stuff, and you should, you know what I mean? You should get respect from that. No.
Yeah, I actually like it. At West Liberty, it's kind of weird. At small schools, sometimes you don't get that laid back feel with the students that you might get at a big R1 school. So it's nice that in this universe, at least, I can be called Doc because
you deserve it and you earned it. Yeah. Mm-hmm.
Yeah. Mm-hmm.
Just saying.
Thanks, man.
Just saying. And I'll tell you, I'm so excited to have you on because I've listened to a lot of podcasts that you've been on where I've gone back and listened to it 2 or 3 times. So why don't we start off just by letting people know your podcast, kind of why you started it, how it's going, that sort of thing. And then we can get into more of like your background and stuff. And it's kind of backwards. I hate that. Like, tell us how you got into reptiles as the starting question. Not my favorite. So
no, I got
if you don't mind, tell us about the podcast so people can get excited about it.
So my podcast is with my friend, Matt Most. I love all reptiles and amphibians. I've loved them all since I was, knee-high to a grasshopper. So, but in my personal favorite group to work with are colubrid snakes. And given that I'm an educator, the podcast environment to me is kind of like the blending of all my worlds, if that makes sense, because you're here talking about something that's very nerdy in a good way. I feel like I'm talking to my people, and I am one of those people. And so when I'm sure we'll talk about this here in a moment, but when my university generated this zoo science major out of the clear blue sky, and then I was put in charge of it and didn't really have the credentials to be in charge of it, I had to go and find a lot of information for a herpetoculture class I was teaching. And long story short, I was trying to find journal articles because that's my world as an academic, and they just didn't really exist. And I was looking for like nitty gritty details, and I started walking around my neighborhood because I got fat and I needed to listen to something. And I found podcasts, and I actually found Morelia Python Radio and started listening to that one, and basically realized that like I was learning an awful lot about animal husbandry and care, and seeing these patterns that kept popping up with some guests, and seeing how some guests were kind of, pushing the envelope and questioning why we did certain things. And so I kind of realized that was an environment that I liked, and podcasts were kind of in this educational space. And then I ended up doing a couple of them, and it was really a natural thing for me to do. And then basically, MPR, Morelia Python Network, they were adding podcasts left, right, and center. And colubroid snakes are my jam. They've always been my jam. They will always be my jam. So I thought I could do an MPR for colubrids. And I knew Matt we have similar interests, but we are very different in our scope. And he's been in herpetoculture literally for decades. I was in it in college, and then I got out, and then I got back in. But I understood like classic herpetology, natural history, and Matt also had that background. So I just thought this might actually work. And so pulled the trigger, bought a mic, and now we have a podcast. And I think people like it. So, that's kind of the origins of that. But Matt and I love it. It is like once every 2 weeks, I know I'm going to be able to just chill out and talk about snakes, snake keeping with somebody that is just as into this as me, if not more. And I'm going to be doing it with Matt. Matt knows more than I do about this stuff. So it's just been a lot of fun, and I don't plan on it stopping anytime soon.
I really think it's a cool show. It's a special show. First of all, NPR, for those that don't know, that's the longest running reptile podcast out, to my knowledge, with number 2 being the Pondcast, the show that you're tuning into right now, which is really cool and something that I'm really proud of, that we're proud of. But it's a great show and a great now network of shows. And I do tune into a lot of them, but yours is my favorite. And it's because of you, and it's nothing against Matt, but I think Matt is kind of like who I am on the turtle side, like one trick pony. Nothing against Matt again, but like he's into snakes. It's what he's knowledgeable about, and it's what he speaks to. Whereas you are more, you're a professor, so you kind of have had experiences, kind of lecturing and working with students, and kind of gaining interest out of people that kind of don't have as much knowledge but are really eager to kind of get in and learn. And then also making a lot of like analogies and connections and things like that with like in life, but also throughout the natural world too. So you're obviously not just a snake guy. You also, there's a crayfish species that's named after you. Like, how cool is that?
Yeah.
And I always want to call you Loughman, but I know it's Loughman. But I just have to say that. So if I say it wrong,
it's okay. Our family's been saying it wrong forever. It's actually Loughman. We went to Ireland and found out that we were lied to, and we thought we were German, and then we did a DNA test, one of the 23andMe, and like, look at that, we're not. So anyway, so yeah, I don't really care how it's said, but it's all good.
One of the cool things, though, like you guys had Mike Rapley on recently, who I think a lot of our turtle people may not know, but he's someone who's been in the turtle game for a really long time and is really well known by some of the people, excuse me, who have been around for a while. And he contributed to my book, actually, and he's somebody who I really enjoy when I have the opportunity to kind of, read something like a post or something or converse or whatever. I haven't seen him in person since 2015, but he's also into colubrids like you guys, and you guys had him on your show. And I think what's really cool about colubrids is that there's a lot of commonality between turtles like that I like, where they don't really need a lot of supplemental heat, and they're terrestrial, and they're just different. They need a cool down, so it's not like those really popular snakes or a lot of popular turtles out there. So a lot of the things that I hear you talking about really interest me a lot. So I want to pick your brain on a couple of those things. If we could just go like topic to topic and just kind of talk about those.
Let's do it.
Does that sound good? Okay. And Steve, you're here too. Everyone, Steve's here. Steve, you're allowed to talk. So don't.
I'll poke in when there's an appropriate moment for sure.
I know you will. I don't want to be that guy who's just like talking and talking.
No worries.
All of a sudden, 35 minutes in, people who are listening to the audio version are like, "What the hell just happened? Who's that guy?" So Steve is here as well, and he's going to pop in. But he always does stuff in the background, so he's a little bit quieter. Okay, so Zach.
Yep.
Talk to me about hormone cascades.
Okay.
That's a thing, right?
So, that is totally a thing. Yeah. So a lot of reptiles and amphibians that live in temperate latitudes, and we need to know what the hell that is. If you're in Ohio, northern West Virginia, Pennsylvania, New Jersey, New England, throughout the Midwest, that's what we call temperate latitude. So basically, where you have 4 seasons. and we're not going to talk about what the hell climate change is doing right now, but classically, you have winter, summer, winter, spring, summer, fall. What has happened with the evolutionary process is that reptiles, amphibians have to have a dormancy period during that annual cycle. It's a literal requirement, because when it drops down like it did here in January to right around 0, if you're a box turtle and decide to go out for a little stroll, you're going to be a turtle sickle. You're dead. Like, that's all there is to it.
Yep.
And what a lot of people think is that when reptiles and amphibians go dormant, that they do exactly what that sounds like, that they just basically shut off biologically and that there's nothing happening internally. And in reality, what's happening is when they're getting ready to go into this in the fall, they've gone from summer into fall, where they had a lot of light and a little bit of darkness with summer. And then as you go through fall, you're getting more and more darkness and less light. Those dial cycles is what we call them of light. They basically trigger hormones, and those hormones are reproductive in origin. And so when we brumate our animals, what people don't realize is that you are basically imitating part of it if you just do the temperature cycling. But if you don't have the light part in there as well, if you're not like dwindling the light as the temperature is coming down, there's evidence that the temperature is far more important than the light. But to kind of really nail it, if you will, you need the temperature and the light. So, but what hormones are being cascaded are in females, they're estrogens. So they're basically all the female hormones, and they're the same hormones that human beings that are female have: progesterone and estrogen, and even testosterone is part of that mix. And then with the males, it's all androgens, which are things like testosterone. So, but what you have to have is, in order for you to have these, in order for you to get eggs in your animal, so we talk about follicles all the time, you have to have this cascade happen with a temperate turtle. Because if you don't brumate them and you're not imitating wintertime, they're just permanently stuck in late spring, summer, and they don't even really begin to do the cycling piece until the fall and the winter shows up. So you're literally going to have them in a non-reproductive state eternally if you don't ever, drop temps. So that's why we do all the cycling stuff. And with our podcast, I decided that I was going to do an experiment. If scientists, what I do. So I was going to go full nerd, and we did that brumation bonanza. And while we were recording it, that one was just Matt and I had PowerPoint slides up, and like my dry erase board was all written up and all that kind of stuff. And what was really interesting, though, is we talked about, the hormone cascades. We talked about how you need to have this cycle. And then a lot of snake people afterwards were like, well, I don't brumate my colubrids and I get eggs. And I started thinking about that, and they obviously are getting eggs unless they're lying. And the number of people that were telling me they were getting eggs was kind of intriguing to me. So resulting of the podcast, I actually have a bunch of Florida king snakes. I went off a king snake deep end here. I did what all herp people do. I was like, I'm going to get 2. And then 10 days later, I had 40. So that's a true story. But I took half of them and just kept them in my office here, actually. And then the other half went in the garage, which gets down to 50 degrees. And sure enough, all they're Brooks king snakes, if anybody here knows what those are. All of the females that I didn't actually brumate all the way, they've mated, and they're getting plump. And all the females that I took all the way down to 50 degrees have mated, and they're getting plump. So it goes to show that when we have them in captivity, we may be selecting against the need to brumate, because if you think about it, you're going to get that one-off clutch here and there, and it might be that one animal out in nature that may have been at the narrow end of the bell curve that didn't really need that much of a cycle for her to go. So
interesting.
Yeah, it's been really kind of fun putting that kind of stuff out there. And that's one of the things that's nice about science. is you can kind of get out of your echo chamber, because my echo chamber was you have to brumate these things. And then I did that, and people said, no, you don't. And rather than be like, yes, you do, and I got my pitchfork and torch and went to my side, I was like, well, what the hell? Let's just see if you don't. And sure enough, you don't with these kind of subtropical critters. So
interesting.
Yeah.
Can I ask a follow-up question?
Sure.
I'll use eastern box turtles as an example, and I've heard this a lot from certain rehabbers. Not to say anything wrong about rehabbers at all. Again, I just like to give that disclaimer, but there's a thought that hibernation or brumation, obviously those are the same thing. I've heard you explain that before.
Yep.
Maybe we can touch on that in a little bit too. But hibernation is a stressor that an animal an animal that's been rescued or can't be released because you don't know its origin or whatever, that they don't need to be cooled or hibernated because that's an unnecessary stressor. Mm-hmm. My firm belief is that it's an important part of their biology.
Yes.
And their natural history. So we should do everything we can to replicate it as much as possible. I think the best placement for an eastern box turtle is an outside pen in a northerly climate where they can be cold for 6 months out of the year and do their thing and live a natural life. But there are people who think the opposite. What are your thoughts, just looking at it from your perspective?
Sure. I'm totally on team cool them down, 100%. And here's why. Even if you're not going to be brumating the animals, there are so many other aspects of their biology that are dependent on that cool down, warm up. Their way to metabolize food. You're going to shorten the animal's lifespan probably, because if you think about it, these animals that are at temperate latitudes, let's just say 3 months of the year, they're not active. So that's essentially like a pause button that's being hit 3 months out of every year. So when the animal is gone, is 4 years old, it's essentially spent one of those years in dormancy. And if you just keep them going at full temp, they will burn out. And there's been some studies that have been published that actually show that happens with some of our temperate animals. You also have a tendency to see stress hormone cortisol will build up over time. And what's interesting is, while hibernation is absolutely a stressor initially, once they get down to temp and they get to that physiological state associated with brumation, the cortisol levels bottom out. And so It's almost like you could think of it as a meditative state. And we're all about meditation now, and you're trying to clear your mind and all that because of the health reasons. And I'm not saying necessarily that they're like zenning out in the bottom of the pond, but at the same time, the cortisol levels it's a different type of stress hormone, and they need that. Evolution is a thing. It doesn't happen on accident. It's selected for, and these animals that were able to live at these latitudes, this is what they do. So my way of approaching their care is we're going to do it. So like at the university, great example with box turtles. As you all, I'm sure, are blissfully aware, everybody and your mother, when they see a box turtle, seems like they grab it, keep it, and then they don't want it anymore. And we have the reptile collection at West Liberty, and a local university had what they actually referred to as the box turtle garden. They put up a fence, they put in 4 box turtles, and then I think it was 20 years later, they had over 20 adults, and they were breeding, and I'm certain there was inbreeding beyond all belief going on in there. And when the university when the guy that was managing it retired, they were going to euthanize the turtles because the DNR said you can't release them. That's not going to happen. So we took on 10 of those turtles, and we have them at the school. And we don't have a place to brumate them, but we're building a building, and I'm excited because we're going to have a walk-in cooler that's going to get down to 38 degrees. And that's where our turtles are going to go. That's where our colubrids are going to go. That's where everything that is at this latitude across the planet is going in that cooler, at Thanksgiving. And then it's going to be coming out around the 1st of March. So
very cool. Very cool. Mm-hmm. Can I kind of alluded to it because I've heard you talk about it before, but can you Can you talk about the difference between hibernation and brumation? Because that's a big one where you'll see
Sure.
people kind of chiming in and saying, "Oh no, it's called brumation in reptiles, you dummy," and it's just another excuse for people to argue with each other online and that sort of thing.
Yeah, that's exactly what it is. It's semantics. 100% truth. Reptiles hibernate. If you say that turtle's hibernating or that snake is hibernating, you're using the correct term. There was a guy at a university in California who did some studies with lizards, and he basically was able to show that when the animals got down, when their body temperatures got below 50 degrees, basically, they underwent a torpor and their physiology kind of stopped. And they got above 50 degrees, their physiology kicked back on again. And the mechanisms at play appeared to be very different than what mammals and birds do. Mostly mammals do. And so basically the biologist said, reptiles brumate, mammals hibernate, and here's why. But then he later on says, of course, that brumation is a form of hibernation. So the reality of it is you're both right. Like it's one of those things, in the snake world, people will say poisonous snake and everybody loses their mind because they're venomous. And there is the one genus of poisonous snakes, but same deal with the brumation hibernation. It's just something for someone to get angry about.
Interesting. See, I didn't know there was a genus of poisonous snakes. Interesting.
Yeah,
there are
learn something new every day.
Snakes. There's venomous snakes. Yeah.
Did you have there's a group of they're actually really closely related to garter snakes and water snakes. They're nature scenes, and they have these nuchal glands on their neck. And when they get stressed out, they put their head down and they present their neck to a predator, and they exude this toxin onto their neck. And it basically, if you get it in your mouth, a predator gets it in their mouth, makes them regurgitate violently. So
wow,
sounds wonderful,
epic.
Yeah.
Has it been tested on humans?
I'm certain somebody's licked it. There's no way.
Florida man, right?
Yeah, there's absolutely no way. Our species loves to do stupid things. There had to have been someone that was like, you know what? I'm going to put that in my mouth. So.
Oh my gosh.
Anyway.
Did you have something else, Steve?
So.
You look frozen right now.
Oh.
That can't be good. That can't be good. Okay, let's just rock with it and hope that everything's still working, even though Steve is frozen. It's going through his computer though, so I don't even know.
Oh, cool.
Yeah. Oh boy. This is awkward. So let's talk a little bit about I had another one. Hold on. Let's talk about the your research with crayfish. I'm really wondering because a lot of them are tied to like riverine situations and streams and that sort of stuff. I wanted to talk about clinal variation. Is that a word or is it clinal? Yeah. Clinal. Okay. I always think I'm pronouncing that wrong too. These are words that I learned from reading. So I always feel like I'm pronouncing everything wrong. So, because I'm not a herpetologist.
Yeah.
So I know Steve was totally smiling, right, when he got frozen. Okay. Somebody in the chat was making fun of him. I love it. So is that something with crayfish or am I making that up?
Yeah.
Yeah.
So when I made the jump from herps to crawdads, the reason why I did that is back when I was in grad school, getting my master's degree, which was when did I graduate? 2002 to 2005. That's when everybody and your mother wanted to be a herpetologist back then. The job market was like crushed. And my advisor told me, go find something else. And so I picked crayfish because everything I learned with herps, I could apply to them because they're huge. They're not like little itty bitty things. And there was a huge conservation need for them. And one of the crazy things I never in a million years thought that I was going to do as a scientist, never thought that this was going to be my job, is I inadvertently got into a situation where I was doing surveys. So I would go into a watershed and collect a crayfish and document what species live there and do all that. And I would invariably end up finding these animals, and I'd have the book that would say it's species A. And then I would be looking at it and I'd be like, you're not species A, but you're not in the damn book either. Like, what the hell are you? And what I was dealing with was exactly what you're talking about, clinal variation. So with clinal variation, what that is you have the core population of an organism, whatever it is, and you have what I always say to my students, makes it so. So I can use turtles. So spotted turtles are a great example. Throughout their distribution, they're a small turtle that's black with the spots on them. And I'm definitely working with Roger Toma. I saw that pop up. He's my mentor. Anyway, and you catch the little spotted turtle up where you're at in Connecticut. Got lots of yellow spots. Blah blah blah. When you come south to where I'm at in West Virginia, we only have a handful of populations, and our spotted turtles are barely spotted. They might have 8 spots total across their body. So what if there was a cline there? What you would see is we would have 8 spots. You would have, let's say, 20 spots up in Connecticut. And then if you made your way halfway down into southern New Jersey or eastern Pennsylvania or even parts of Maryland, if they had 10 spots, then what you have is this thing we call a cline, because you are gradually going towards a different phenotype geographically. So what happens with the crayfish is you get into these stream systems, you pull the crayfish up, and they show a lot of what we call morphological plasticity, which is really fancy words for the claws are not the same in every river. So then you have to sit there and ask these questions to yourself, which is this a new species? Is this not a new species? Is this a random form of the phenotype of the species that we would see over in the drainage system next door? And so what I ended up having to do, and I didn't know I was going to have to do, was learn how to name species. And that was not something that I intended on doing. What's really ironic about this is one of the reasons why I got out of herpetology is when I was getting my master's degree and I was looking at PhD programs back in 2004-5, there were basically 2 things you could get a PhD in herpetology back then, that where you would be funded and you wouldn't be starving. And one of those things was phylogenetics. So you're going to basically be the gene jockey that goes out into nature and catches 50 whatever, frogs, salamanders, turtles, snakes, takes some tissue, brings it back, runs a DNA gel, and then makes a phylogenetic tree and says new species. And I didn't want to do that because I'm a classically trained ecologist and natural historian, and the gene tree peeps generating cryptic species that you couldn't, like, you could tell apart with DNA, but you couldn't look at it and tell it apart. Right. I came out of a lab where that was like blasphemy. So the fact that I do, that I had to go and describe species and do all that kind of stuff. Now it's very interesting that I ended up being what I didn't think I was going to end up doing in the first, ultimately.
That's really, really funny. That's really funny. Yeah. I think this is a good kind of segue into like the idea of like lumpers versus splitters. And I think a lot of people in our world, in this turtle world, will say like, oh, this is different, it should be a different species, or is this something special, or is this that, or whatever. And there are people who feel like, and I think there's different situations, like the European pond turtle is one where there's a whole bunch of different subspecies. You can't tell most of them apart morphologically, so then you're kind of stuck there. You need to do genetic work to know what they actually are. And then sometimes I think the science lacks sufficient sample sizes to really know. And, maybe there's a paper and some people dispute it, but we're not really sure what actually is coming out of it, or how widely accepted it is, and how many animals even went into describing something. And I know there's one where incubation temperatures can change the way they look. Okay, great. So it looks like this species because the incubation temperature, like what is actually at play here? And it's really tough to know. And then there's this clinal situation as well, where you have a species that can, and we've talked about this on the podcast before, but you can have them at the top of the river system, and then way down at the southern part, they can look totally different. And when you go out there and you take those animals from both spots, at the top and bottom, they're completely different morphologically and genetically, and you don't realize all of the steps and all the gradual changes all along the way that goes into that. I think Eastern painted turtles are a good example. Like basically everything is an intergrade zone with painted turtles. You pick one up, it looks like two of the subspecies. But like, so where are the actual, type specimens, and how much space do those type specimens actually make up? Because they're all just mixed up. And then there's people moving around and everything else. So sorry, going on a little rant here, but I mean, your thoughts on the 12 things I just mentioned.
I think that we're actually in a really good time to be a taxonomist, because it took about 25 years for the old guard of taxonomy to accept that DNA is not going away. It also took 25 years for the DNA people to lose their arrogance and completely disregard everything that the old guard had done. And now we're kind of in this happy Goldilocks zone where there's a new species concept. It's been around for a decade, but it's definitely the concept that seems to be bearing the most weight across all taxa: crayfish, birds, snakes, turtles, whatever. And that's what we call the integrated species concept. And what that concept does is basically you use you use evidence from multiple lines. So you have genetics, you have geography, you have morphology, and you kind of look at what the whole picture's saying. And once you have that, you can move forward and we're good to go. So that's what we're doing in the crayfish world. I've read a couple snake papers recently where I was like, okay, cool. This integrated approach is starting to be used because the reality is there's so many different lines of evidence, and you like the genes. Let's talk about the genes for a second. For the longest time, when the gene approach showed up, a lot of people were like, oh my God, this is a game changer. And it kind of was a game changer for certain taxa. But for other groups of animals, it was not a game changer. And actually my group, crayfishes, were one of those groups. The barcoding gene, this gene called cytochrome oxidase 1, it with crawdads made one species like 30. I mean, it was crazy how diverse this gene was. And the reason why it was being used is we use the genes when we're doing molecular taxonomy that are going to be what we call evolutionarily conserved. We want a gene that isn't going to mutate all the time. So that if you get a little bit of mutation, that little bit means something. And that's why those genes were used. Now, it's 2022. Those studies were being done in the 98 through 2005. Now we can look at the whole freaking genome. And it took forever for us to do that with human beings. I just found out that one of my colleagues with crayfish looked at figured out the genome of a crawdad in his genetics lab using college sophomores. Like within a decade, just sit back, because one thing I will say is that I know people get a little bit pissy about the taxonomy piece changing. It has not changed yet. We have not seen the end. Because we're getting to the point where we can literally look at every single gene. And so we don't have to choose and have that subjectivity of which one are we going to use. Within the next decade, we're just going to look at the whole damn turtle. If you know what I'm saying. And that'll change everything.
Right. I mean, Brad Schaefer at UCLA has been working on a lot of this stuff, as I recall. And like, I'm really looking forward to some of the things that, come out of his project. I've gotten a chance to check out some of it, and a lot of it ironically suggests, maybe not ironically, that, some of the stuff the old guys used to do, was completely accurate as well, which is really cool to think about too, is even now, as we can like build the entire genome, we can be like, oh yeah, the way they thought these pieces put together, they were right. And obviously I'm as interested in it for the map turtle stuff as anything, because there's especially in there's always been this kind of groupers versus splitters thing sometimes.
So I'm really looking forward to seeing some of those results and seeing how they blend with what we know, with the morphology and all these other things that kind of go into the taxonomy piece. I mean, in map turtles, you even have the fact that they've been isolated in separate drainage systems for how long,
Yes.
and how long that's going to continue, that you have to take into account, and not just the genome and the appearance and all these other things. So like all those other factors are kind of cool to look at too.
Oh, 100%. And the old people, I love how I would say the old people, but the originators that were doing the taxonomy, they're looking at morphology. And what controls morphology? Genes. So it makes total sense that if you look at turtle A, turtle B, and you're like, well, these two are different morphologically, that then when you look at them genetically, that there might actually be something backing that, because genotype rules your phenotype. So
I think the trouble comes in where we were talking about, like, what happens over the entire range.
Yes.
And because if that's missing, then it becomes really problematic. But then also, what's the sample size? Because if you have 5 over here and 12 over here, and they look different, you're leaving yourself open to not really knowing exactly what's happening in either population.
yes.
And that's what I see in some of the turtle stuff, especially with some of the rarer stuff. I mean, unfortunately, with some of these species, we're trying to figure out, the taxonomy and where things fit together when they're already extinct in the wild or critically endangered and that sort of thing, when there's already been this huge effect by humans on these populations, which is where, the turtles really differ from some of the other vertebrates and reptiles as well, in terms of their situation and how important this stuff is, but also how messed up and convoluted and confusing everything can end up being.
Yeah.
So, yeah, it's cool, but yeah, Chris, you know where my head's at always. Talk about those Asian box turtles always. I don't stop thinking about those.
Yep.
Zac, can you tell us a little bit about your program and what you're doing at West Liberty?
Sure. So I teach at a small liberal arts school in northern West Virginia. It is basically where we're located is the northern panhandle. So West Virginia has an eastern panhandle, and then this little part that sticks up between PA and Ohio. And that's where my university is. And that's also where I went to school. I grew up here. I actually teach in the building I got my undergrad degree in, which is pretty awesome. But when I went back to West Lib to teach, I slowly but surely built a small environmental program there. And I had my crayfish research lab, and we were growing, and hired a professor that was one of my friends. There's a local zoo in Wheeling, West Virginia called Oglebay Good Zoo. And during college, I worked at the zoo. And then this guy named Joe Greathouse worked at the zoo with me. And so he went off and got a PhD. He studied hellbenders. And I wanted to bring him on because I study crayfish. He studies hellbenders. We could get a freshwater conservation thing going. And he had been in AZA forever. Like, the zoos were his life. And so when we got him hired as a prof, he started talking about this thing called a zoo science major. And I was like, well, what the hell is that? Like, that sounds interesting. And basically the idea is that while you're getting your undergrad, get all the experience you need to get a job in AZA. And we all know, like, it's not a secret that keeper jobs in AZA do not pay that great. But what this basically gets you ready for is kind of that lead keeper, assistant curator gig. Because you have to have the knowledge, you have to have the experience, and you have to know AZA. And so that's basically what this degree would do. So back in 2015, we started putting this whole thing together. And the idea initially was that I was going to run the ecology side, and Dr. Greathouse was going to run the zoo side. And lo and behold, he was too good at what he did as a zoo guy. And so he came back home. We got the major set up. We got the relationship with Oglebay going down. And then he got offered the director's job for Oglebay Good Zoo and bounced. And they're like, oh, by the way, Zac, you're in charge of zoo science. And I was like, excuse me? What the hell? I don't know. Like, I was a part-time keeper with goats when I was an undergrad. I cannot be managing this major. But I'm also the kind of guy that likes a challenge. I really like to jump off of hypothetical ledges and have absolutely no idea how I'm going to land, but I'll figure it out while I'm falling. And I knew that this was going to be a good thing for the school. So I thought, what the hell, I'll give it a go. So in 2016, we started this thing. And what was really interesting is I told all the people that I worked with, like, we're not going to half-ass this. This is not going to be done where they go do an internship at a zoo and that's it. Like, we have to have a zoo here. And so what we ended up doing is got our own animal collection. And so I was into herps and herpetoculture and all that all through college, all the way up until about the time my son was born in 2008. And then I got out because I was getting my PhD. I had a child. I did not have time to give the animals justice. And this was crazy. Like, this was literally like somebody that has, kicked their cocaine habit, and then their boss comes up to them and goes, "Hey, you want some?" Like, literally, that was what this was like. So I basically had the keys of the kingdom, and I built this herpetoculture laboratory now. And we, in 2016, had a corn snake that I always had at least one animal through that whole period of time. He lived in the zoology lab, and as of a month ago, we have 370 animals at the university, and all major lineages of reptiles and amphibians are covered, minus we don't have a tuatara. So there you go. But we have plenty of turtles. We have crocodilian, smooth-fronted caiman, and then lizard snakes, salamanders, frogs, toads, hylids, the whole kit and caboodle. And then everything that we have there, I started like networking with curators at zoos because I wanted to make sure we set things up right. And then, as you can see, I brought it home with me. So now I have the animal collection here. And then my house actually serves as the quarantine for the zoo side collection because anybody that's listened to me on podcasts has heard about a crypto scare. So I just put everything out in the garage. But no, it's been crazy.
That little comment right there gives us a perfect segue to one of the questions that was asked.
Yeah.
You said your house is the quarantine environment. And so we had a question already. So what's in some of those things that are behind you that everybody can see in the racks and on the other side?
Okay. So let's see. Well, back here, those are rhacodactylids. Those are what's in there? Those are some crested geckos that I raised. What's funny about those is those are actually bioactive vivs, so they're fully planted and all that.
Nice.
And I love the geckos. I love the plants just as much.
Yeah.
So that's what's living back there. And then in the rack over there, the top of it has a bunch of Madagascarophis colubrinus. What are those things? Madagascar cat-eyed snakes. And then the bottom are all dipsadids. So I'm writing a book on false water cobras and musaranas and Barron's racers. And that rack has a bunch of these really weird little colubrids. They don't have an easy name. Erythrolampris, Postilla-gyrus. I think they're called yellow-bellied Liophis. They're like little garter snakes from South America. And then I have you can't see it here like that, but there's like a bank of PVC enclosures that make up the wall. And so that false water cobras and then the king snakes and Nerodia. So yeah, lots of stuff in here.
Lots of stuff. Are you using racks for all ages or just for certain life stages?
I use racks for snakes that don't do well in PVCs. So I like naturalistic enclosures just because I like to see the animals. But I will be the first one to admit there are definitely animals that when you put them in these enclosures and they're exposed and I come in here, they freak out and they go off food and they don't do well. So everything that's in a rack is either growing up to ultimately go into the PVCs or literally just want to be in the damn rack. So like the Madagascar cat-eyed snakes are a great example. I had them in the bank of enclosures right here and they just stopped eating. And so I thought, okay, I'll put you back in the rack. And then they ate, and then I put them back into the enclosures, and they stopped eating. So they're in the rack forever.
Interesting.
But
I have some theories. Go ahead.
Go for it. Oh, I don't have problems with racks. You can do animals justice in racks. you're not going to be providing them light, and there's absolutely a potential issue there, and I won't negate that. But for things that are crepuscular or extremely nocturnal or fossorial, I almost feel like my experience has been that they've done better in that system.
So that's a big one on our end with the turtle stuff. A lot of species early on are fossorial, or like their biggest. I wonder about cortisol or corticosterone levels or whatever. Like that they spend their entire lives for the first 2, 3, 4 years completely hidden under leaf litter, under dirt. And, we take them, and then the first thing someone will tell you when you're trying to set them up is you need to have UVB. And then you set them up in these like bright boxes, this glass case of emotion, and they're supposed to do well. Like I worry about that a lot. And I actually use racks myself for small box turtles and other like terrestrial turtles when they're small, and a few pond turtle species as well that aren't like avid baskers at that young stage. I think a species that wants to bask is one thing, but one that wants to hide is a completely different thing, especially at that life stage. So that's what I was asking. I also wanted to ask, so you're talking about herpetoculture, and for anyone who hasn't heard that term, I'm sure most of the people who are listening have, but that's the different. Herpetology and herpetoculture is the difference between academia and a hobby. And the fact that you actually do you have courses in herpetoculture, or do you actually have a major in herpetoculture, and what level is that major?
We don't have a major.
Okay, that was my understanding, and then I got to
with the zoo science major though, and we have a graduate degree. We're the only I can't say this, and we're very proud of this. We're the only zoo science graduate degree in the world that we know of. There's no other one, and it's completely online. Shallow plug. Anyway,
make the plug because this is a new audience who may not have heard you before, and it's super duper cool.
But we offer courses in what are called tetrapods. So all the animals that have legs that are vertebrates are collectively known to people like me as the tetrapoda. So that includes birds, mammals, reptiles, amphibians, and then the fish are the other group of vertebrates. And with zoos, those are the main groups that people work with, and they're kept in zoos. And then if you have the fish, obviously, that's aquarium science. That's a whole other can of worms. So we offer a semester-long course for the undergrads and the grad students in mammalogy, ornithology, and herpetology. So I teach the herpetology class. And the way that those classes work is the lecture course is exactly what you'd get anywhere in the country. It's evolution, biology, taxonomy, systematics, ecology, behavior, like all that kind of good old school applied natural history. And the lab is where things differ. So a traditional ology class, you would usually learn to field ID the animals that live near you. So like I took herpetology twice. I took it in undergrad, I took it in grad school. And both times I learned the reptiles and amphibians of West Virginia. So in lab with a bunch of dead herps. With our class, or with my class, we don't do that. We do herpetoculture in lab. So like I have a lab, it's a 15-week course. So there is a 4-hour lecture on lights. And then there's a 4-hour lecture on cages. There's a 4-hour lecture on substrate types. And there's a 4-hour lecture on habitat heterogeneity. So like naturalistic versus bioactive versus sterile. When do you use which? What's the difference between naturalistic and bioactive? How do you use all this stuff? We do a veterinary thing. And then there's dedicated lectures specific to the herpetoculture of turtles, herpetoculture of salamanders, herpetoculture of frogs. So it's intense. And the grad class is like the undergrad class on steroids. So with the grad class, what I'm doing, it's actually my favorite class I've ever taught, is you do all that same stuff, but we basically read 3 journal articles a week on a topic. And then there's a forum question, and then you have to respond to the forum with these journal articles, and then you have to respond to 3 of your classmates' responses. And it's kind of fun because right now I would say about 50% of the grad students are herp nerds. So they're like this is a class. I'm doing this for a class. Like they're like in shock. And then there's like, I want to study bunnies. Like there's mammal people in there too. And so it's kind of fun to see the like whole interactions. And at the beginning of the class, the students are like, this is too much work. I usually have to shut down a coup. And I basically say, you have to do it. And what's really funny is about 3 weeks in, I've done this class now 3 times, and everybody's into it. They're nerding out. It's kind of a fun environment because you learn how to debate, not flame. So like when you go on social media and people were like, well, I keep my turtle this way. And then someone's like, well, that's the wrong way. You learn how to, in this course, be like, well, I keep my turtle this way. And then somebody says, well, I keep my turtle this way. And then you try to communicate to ultimately figure out how to keep the turtle the best way. So that's another part of the class, is just, doing that. But I'm really, really big. I'm a massive proponent on, you got to understand the natural history of the animal before you can keep it. So what I do is I'll find like the life history of the Eastern box turtle and make people read this monograph from like 1890 or 1920. And then I'll say, okay, you've read the paper. Now, how would that change your keeping strategy? Or I'll ask a specific question. So like you read the paper and then I'll be like, all right, so what's the lighting regime need to be for this turtle? And how is that going to impact its breeding? So I'll pick like a specific facet. So I'm just trying to get people to think, because I learned in my second coming as a herpetoculture person that before, when I was just doing the stamp collecting, when I was in college, I want this, I want that, I want this, I want that, I want this, I want that. There was no meaning behind it. Now, a lot of my snakes are little and brown, and people would look at them and be like, why the hell did you keep that? Well, the biology of this thing is phenomenal, so that's what, that's the driver now. So, but no, the program's working. Our undergrads are getting hired. We were scared to death with COVID because the zoo world took a huge hit, because zoos are gate driven. So when you shut down people going, you're going to have to lay off people. And we thought that the job market was going to get saturated with all these keepers that had all this experience. And what we've actually found is that our students are getting jobs fairly, like the ones that put in the time and do it right, they're not having to worry, and they're getting like really freaking badass jobs. Like some of the jobs my kids have gotten, I'm like, well, I want that job. So no, it's definitely working. And the grad program, the problem we have with the grad program is the grad students are getting employed in zoos while they're pursuing their degree, because they're in the degree, because the zoo's happy they're going to have a graduate degree, but then they get a job and they're like, well, I got the job now, so I have to like do everything shy of threaten them within an inch of their life to get them to finish. So anyway,
it's a good problem to have, right?
Yeah. Mm-hmm. But like we're at 100% employment rate. So you can't, with the graduate students, every single grad student who has applied to AZA has gotten a job. So you can't beat that statistic.
That's really awesome. That's a tough racket to get in as well. Like, I mean,
it's competitive.
Of the couple AZA institutions here in Connecticut, like a lot of people who I know are just, volunteer for 10 years if you want to get a job in the zoological field. Yeah. That's really that. Thanks, Chris. You're right. That's really cool. Took the words out of my mouth.
So,
wow. And I mean, you're looking for students as well, so people are interested, right? So you make the plug. Like,
I'm always looking for students,
like online for the graduate stuff, and I mean that's so cool. Someone just asked how many students graduate each year in the zoo program. Yeah, I see how many students graduate each year. We're averaging somewhere between 20 and 30, and what happens with this major is a lot of for the undergraduate degree, a lot of people think they want to work in a zoo and they have no experience with animal husbandry. And anybody that has buku experience with animal husbandry knows that you are cleaning poop, you are going to smell, you are getting a degree to get
you're getting a degree to get salmonella. It's going to happen. So like, but what we realized is about 30 to 40% of the undergrads about the end of their sophomore year are like, well, I don't want to do this, but I want to do conservation. So that's actually where my like background is. I am a conservation biologist. So we created a track in the major so that at the end of your sophomore year, you have to declare you're either going to be a zoo science major or you're going to be an applied conservation major. And you don't lose any time. It's a very smooth pivot. And then you're just basically getting a degree in field biology is what it boils down to. So, and what's really cool is we've been able to like show the students how captive husbandry and field biology are intimately linked with things like head starting and repatriation and purchasing
awesome.
habitat and all that kind of good stuff.
Right.
Right. Yeah. And coming from the educational field, like to be able to design your program where they can make that turn and not have to go back and redo stuff or whatever. That's what we call quality design in education. And like that's something that like we talk about, like even in our high school is, okay, so a kid was advanced in 7th grade math, but then they struggled a little bit back in 8th grade. How do we give them, how do we let them, parachute down a little bit? Or somebody was, on level in 7th grade, but all of a sudden they've sped up. How do we allow them to move back up and make that change without having to necessarily like just suffer behind or any other thing? So like, it's really cool to see that kind of intentionality in the design program so that you can really get kids interested and even send them off in a slightly different direction without having to feel like they need to backtrack and just shell out some more dough. So that's really cool.
No. One of the nice things about my being at a small liberal arts school is that we can pivot.
Yeah.
And there's bureaucracy. I am not saying there's not bureaucracy. But we're kind of able to bully the bureaucracy, if that makes sense.
Nice.
And then the zoo science major has been so successful. It's it's either the second or the third largest major on campus right now. So
wow,
because
that's incredible.
Of our numbers, the upper administration has basically been like, okay, don't know a damn thing about all those animals over there, so we're just going to let those people do what they're going to do.
Right.
And that's
but I mean,
nice.
Do you find, are you pulling people from across the country at this point? Because of their interest. Like that's the really cool part. I mean, how many majors are doing that at West Liberty University, right?
Yeah. Oh, well, we're the only one.
Right?
Yeah. The only one. That has our dental hygiene is but the biggest issue that we have with the major is the simple fact that people don't know it exists. Because when you're at a small school, you don't have the marketing budget that you would have at a large university. And that was also one of the reasons why I did the podcast and on every podcast I can go on because I know where my target audience is. It's I am the target audience. So I just basically sit back and think, well, what am I consuming? Right. And it's totally done it. And hey, if it gets a bunch of reptile people that I get to teach, damn. I'm so upset that that's the dominant.
I love that you're the target audience. So, it's like you're going to a holiday party and there's going to be a Yankee Swap or like Dirty Rotten Santa or White Elephant or whatever the heck it's called. It's smart to don't re-gift a candle that you didn't want. It's smart to bring something you want because then people are also going to want that. But I think that's so funny. Like that's who you are, and the people that you're trying to attract are the same.
Yes.
Is 72 years old too old to start?
Heck no.
No way.
Why not?
I saw an article with a guy who was 72 who was playing college basketball and shooting free throws.
Yeah. Let's do it.
You could definitely do college herpetoculture and zoo science if you can make it on the baskbasketball team.
Yeah, we had a woman that age in our college choir where I went to school. She was she was just picking along courses in her 60s and 70s and just kept singing in the the not auditioned choir, but the other choir just every semester. That was one of the things she did.
I love that. I love how you said the non-audition choir. You got to make sure keep
right. Well, yeah. Well, they. you didn't have to audition for the college one either. So like anybody could join it. And so she did. It was, a thing she did. So
That's cool.
yeah.
Nice.
Ryan says his girlfriend has lots of coworkers showing up thinking they're going to just cuddle kittens and puppies as vet assistants.
Oh yeah.
I feel your pain. Your pain.
We call them bunny huggers.
Right.
And the stories that I can tell. So one fun one is we had the first semester in, we had this girl and she bailed out of the major and was like just really angry one day. We didn't know what the hell was going on. And then the next day, somebody from HR like reached out to me and was like, "Hey, we got to talk about what you're doing with those rats." I was like, "What do you mean? What am I doing with the rats? I don't know what you're talking about. I don't do anything with the rats." She's like, "Well, you're drowning the rats. And the kids don't like that. Like, we can't be drowning rats. That's cruel." It's like, we're thawing them out. They're frozen. This girl thought that we had these rats somewhere on campus and that I was driving up in the morning and just drowning rats by like the tens every single day. And I was like,
oh
no.
My gosh.
Like, so there's a very interesting dynamic when you have a major like this of we get students who literally like eat, breathe, sleep aviculture, or herpetoculture or tropical fish, and they know everything coming in. I mean, they're like rock stars. And then we get other students that have watched Animal Planet, the zoo shows, and think that all they do is that. And there's nothing wrong with those students. Some of the absolute best kids we've had are those people, and it's amazing to be in a position to like teach them how to do all this and watch them, start naive and end up being these wickedly experienced, wonderful future conservationists. I mean, it doesn't get any better than that. But it's been there have been things with this major that I was not prepared for at all. And it's been a lot of fun, and it's been a massive learning experience. I can absolutely say that.
So I think there's a question that's been asked that ties into this a little bit. He says, I'm sure Zac knows about graphic care sheets floating around on social media, Facebook groups. They claim here is the only way to care for a particular animal. Do these discussions show up in your classes, and what are they like?
They absolutely do. And we actually have an activity in the herpetoculture class, which I tell them to go out and find the care sheet. And then I tell them to then go out and find the journal articles that discredit the care sheet. So I do
I like it.
An exercise, and there's more than one way to skin the cat.
Yeah.
Because that's one of the things that I want to make. If I have any impact on herpetoculture, I just want people to not do the care sheet thing, and be so defiant, like not defiant, but to honor it with like their life and blood, like
so rigid.
Rigid. Yeah. That's it. Because I've heard this on every podcast. When you get experienced people, the way that I keep things in West Virginia is going to be totally different than the way you do it in Florida, which is totally different than the way you do it in Montana, which is different than how you do it in Holland. Because the environments are different. So if you get that little care sheet and that's what you're basing everything off of, it should be, I teach the students it's a foundation, but it's not like the end all be all Bible of care.
Right. Yeah.
Right.
So.
I think, one of the things I think about when we've written some care sheets, at the Turtle Room, one of the things I think about them is it's kind of like, what do the principles look like? You don't need to detail the habitat should be 8x4 with X, like the care sheet should be focused on principles of needs and all these other things, and then leave it open to some interpretation of how that happens in wherever you happen to be. Because like you said, like those of us in the Northeast, we learn how to do some more stuff inside that you can do outside in Florida. Those folks in Florida, they get stumped on certain things because they can't all of a sudden keep them outside because it's either too hot or whatever else in Florida. And they're like, I don't know how to do anything inside. And so like we gain different areas of expertise just based on our region as well.
Yeah, 100%.
Sure.
No,
that was something I had to realize. I really struggled with it. Like, I'm going to retire and move to Florida, or I'm going to move to Florida as soon as I can because then I can keep so many other things. But there are a lot of species that actually do really, really well in a cooler climate, whether that means you have a basement that naturally gets into the 50s in the winter, or, you have animals that really do well outside here year round, which is crazy to think, but they are out there and finding the right species for you. Go ahead, Zach.
No, I agree with you 100%.
Yeah.
It's funny when I got back into this, because I'm a snake guy, I had all these dream species that I wanted to keep when I was in college. And so I thought, okay, well, we're going to do this. And it's funny, I've gone like full circle, and now with the personal collection here, it's just a whole bunch of freaking king snakes. Like that you throw the mouse in, they eat it. But I like the phenotypic plasticity, and I like the evolutionary story there, and the locality stuff's really cool to me. So yeah, I did see a question that I wanted to talk about.
Please.
I don't know how to go down in the chat, but somebody asked me what I thought about the private sector.
Yeah, that was Andrew.
Yeah,
who also made fun of my vest, by the way. Thank you, Andrew.
Yes.
I got out of work late today. What I wore is what I got. I'm embarrassed. I look like Aladdin.
Yeah, I like the vast. I am not the guy that poo-poos the private sector. There are people in the private sector, obviously know there's aspects of the private sector that we just don't want to talk about. But there's definitely, I think that there's a lot of potential for the private sector to do a lot of good. It's just getting to the point where we're allowed to do it, if that makes sense. What's fun about my position is I work in academia as a professor, and then with the crayfish, I work with Fish and Wildlife. I'm a species expert for 2 endangered species. So I am hip deep, ear deep in the Endangered Species Act at the federal level all the time. It's never ending. And then I work at state level. And what's fun is in those circles, I'm the crayfish guy that works with snakes. And I've heard, I've brought up snakes and regulations and why do you do what you do and all that kind of stuff. And I will, people call me naive for this, but I think that there is a way to kind of reach the happy middle land where everybody can kind of have their purpose. And I do have to say that I think the turtle people are the best at it. Because snake people are, not to use generalities, but we just have a tendency to be very angry. So the second that someone tries to take something away, there's yelling and screaming and there's no diplomacy. But I definitely think that the private sector has its purpose.
I think that's actually when I first had my man crush for you grew at the first moment. Sorry, that's totally inappropriate and weird, but just being honest.
Okay, man.
But
it's Monday, man crush Monday, right?
I'm sorry,
social
it is what
media
it is.
thing, right?
But you were on a podcast and you mentioned that you felt like the turtle group, in terms of herpetoculture, that the turtle people did the best job of kind of trying to put effort into and branch out towards some sort of semblance of conservation, which we all know that like true conservation is like a unicorn or a leprechaun. It's not really something that you see that often, but especially coming from the herpetoculture side, but we try it a lot, and we at least get into like preservation and keeping species for the right reasons and all that sort of stuff. So that was one of the first things I heard you say, where I was like, wow, like this guy, he loves snakes, he loves crayfish, but he's got a really good idea of everything if he also is into the turtles and the other stuff, which I thought was really cool and unique.
Yeah. And I'm like, even with the university, I'm trying to get in on head starting programs, and I want to use our space because we have a budget, and it would be an amazing opportunity to teach my students how to raise things that are ultimately going back, and we can support that. But it's been such a climb uphill to get anybody to commit. But I will say this, even me as the endangered crayfish guy, like that helped get these 2 animals listed as a species expert, when they were listed, the recovery plan that came out for these animals said there were 2 things we had to do to get them off the endangered species list. We had to restore streams, and we had to rear them in human care. And I already knew how to rear them in human care because it's really easy. They mate. The females retain a sperm capsule and spermatophore, and then they basically overwinter with that. In the spring, it warms up. They fertilize their eggs. They got eggs. So and you got maximum heterogeneity too, because they have multiple baby daddies. So I've been trying to get Fish and Wildlife to let us do this. And just so people know, it's not just hobbyists that get shut down, because I have to jump through so many hoops to prove that we're not going to be causing take, which is killing the crayfish. And we have to use surrogates. And then we use the surrogate species, and we show, and we get to like the final point. And I'm like, all right, yes, they're finally going to give us the permission. And then we get hit with, well, have you ever done it with the target taxa? And I'm like, well, you didn't let me do it with the target taxa. Well, we need to go back to the beginning and now slowly do it with the target taxa. So conservation, it's a slow crawl. And, it unfortunately, it needs to be a sprint. And you're looking at a guy that despises bureaucracy. I hate it. I loathe it, because it's what leads to extinction. But at the same time, I think the private sector oftentimes thinks that there's a target on their backs and that they're being discredited and they don't have any say. And in reality, the academics of the world have the exact same target on their back. And I don't think people realize that.
That's really interesting and really valuable to know.
Yeah.
Wow.
Like I have a building that we're building. I got a big grant to build a crayfish building and aquatic conservation center. And what's interesting is the building's being built because Fish and Wildlife doesn't have anybody that can rear these crayfish, because they don't want to kill them. So I got a building to rear the crayfish, but it's going to take a year to build the building. And then when the building is built, I then have to apply for the permit to put the crayfish in the building, which is going to take another year and a half. So like right now, 2.5 years from rearing a single crayfish in there. Meanwhile, the streams are being polluted with coal waste in southern West Virginia as we speak.
Right.
So
of course,
and where are we going to put the ARC population? So like all these things we talk about, I sit back and I hear the conversations, and I just want people to realize that that's just part of the process. Zoos have to go through the same thing.
Yep.
But the turtle people, y'all did a great job with convincing people that it was worth, you guys doing what you do with the turtles and spotted turtles and diamondbacks and all that kind of awesome stuff.
It's a challenge. Like in my home state, it's much more of a challenge. So the good thing about being kind of a larger organization now with a larger scope and reach, I guess, is that you can kind of figure out where are our significant players on the biology, the field work side of things, and then where are the states that are willing to work as well, because every single one is totally different. And if you find yourself in one of those states where people aren't willing to really let you be creative and try to help out or come up with something, then it is what it is. Yeah. Last time you and I spoke on the phone, it was a while ago now, but we spoke on the phone, and at that time you were interested in potentially taking some turtles into your project. Now, turtles aren't the easiest thing to keep. Are you still interested in that?
Well, we're totally interested. When this building's built, I have an extra aquatic life lab. That's what it's called, because I didn't want it. I wanted it to have reptiles, amphibians, and turtles. So I'm currently talking with the West Virginia DNR about some things. I don't know where that's ultimately going to pan out, but they're wonderful people. I can say that our non-game people are good. But I've tried to get in with PA and, all that kind of stuff. So we're always willing.
Best. Yeah. That's
we
awesome.
can raise things.
That's
and
great.
We have a budget. We have a budget to cover food. So
I love it. So we look forward to partnering with you as well. Like however we can.
Yep.
If you want a guest lecturer to talk about turtles with your group, just
done deal.
Let me know. I got really excited when you were talking about herpetoculture of turtles, or however you phrase it. And if anyone has something cool that you'd want to see go towards education, I mean, this can actually go to the new facility there and actually be a part of that education for students who will be, doing research and I mean, like your grad students. Are they publishing papers and things like this on this sort of thing?
Absolutely. Can
you give us an example of like a cool one?
Sure. We actually had one accepted today. So
awesome! Congratulations.
Yeah, my graduate student, Michelle Williams. So one of the things that I really have gotten into is reptile cognition. So these things are not little androids running around. They definitely are associative learners and all that kind of good stuff. And so we did target training with false water cobras, which are not real cobras, in case we don't have snake people here. I wouldn't do that with a grad student. But we were able to show that they could basically learn a target and learn what the target meant in as little as 8 sessions. And so we did this for zoos because, you don't want to necessarily go in and grab the snake and have it bite you. If we do this technique that Michelle developed, you can present the target, move the animal to a shift box. It willfully goes in the shift box, gets rewarded. And then you can go from there. So that's one. We have another paper we submitted last week, which was so corticosterones in poop. That's a way to non-invasively test for stress in reptiles and amphibians. Mm-hmm. So like when you go and do a blood draw, they get stressed, and then that's going to end up in the blood when you pull it out. And so you end up not really knowing whether the animal is permanently stressed or not. And there was some debate with squamates that the mammals they were eating were stressed, that corticosterone was passed through the food into them. And so they were then pooping out stress hormones that were not necessarily their stress hormones. So I had a graduate student, joys of being a graduate student, take snake poop, cook it in an oven, get all the moisture out of it. And then we ground it with a mortar and pestle and then shook it through some sieves and got this like pure fecal powder. And then the bones and the hair and everything. And we did corticosterone levels of the powder and then corticosterone levels of just a normal poop. And we were able to show that if you do that extra step and get the poo, you get lower cort levels. So basically, it kind of demonstrated that if you add this one step, you can actually get a more a cort level that is probably more reflective of the actuality that's going on there.
Right.
But then I have
so
others.
Cool.
I have other students though. So many people might know Lucas Lee. Lucas Lee he's part of the NPR group. He does Carpets and Coffee, that podcast. And he heard me on a podcast and came on to be my graduate student. And he loves Australian pythons. Like that's his jam. And we were trying to figure out what we were going to do for his thesis. And it was COVID times. And he said to me, he doesn't know what he did. He messaged me over Messenger, what if I just wrote a book on womas for my thesis? Ha, ha, ha. And I was like, what if you write a book on womas? So now Lucas, for his thesis, is going to be the lead author on the complete Woma Python, the complete series from Eco.
Yep.
So he's doing the natural history, the evolution, and captive husbandry. That's for his thesis. And then we're going to take those chapters, and then Nick Mutton and Dr. Julander and I are going to be on it as well. But like that's the kind of thing we can do with this graduate degree. So
right,
if there are turtle projects that you have the data sitting there and you've always needed that one final step, that's where grad students are wonderful creatures, because you can give them the data and they have to you have to get that final report, document, paper, pub, book. Now, obviously, the kid ends up being the lead author on it because they do all the work, but it just kind of helps everyone. And that was kind of my goal with the grad degree, was I thought that this was a great way to kind of segue really dedicated keepers on the private sector into the world of academia and get them into AZA. And then you have somebody who's literally a keeper, an academic, and an AZA, and you kind of have a trifecta there. And the more of those people we have, the better everybody's going to be.
Yeah, especially for research on keeping, various different parts of the herpetoculture, as opposed to just, kind of the ecology or biology too.
Oh yeah, absolutely. And there's plenty of cool stuff. So I love the grad program. It's I listened to this guy named Warren Booth, Dr. Booth. He's a professor in Oklahoma. And he does a lot of parthenogenesis research with snakes, but the thing that pays the bills with him is the genetics of pests, and he referred to it as hobby research. And I was like, yeah, that's kind of what this is. Like, it's like how I just think I'm the luckiest guy on earth that I get to do this every day.
Right.
Like, how did this happen?
It's so cool. And I love the idea of like the different branches that come off of the majors too, and how I envision these students who kind of are growing up having an appreciation for people on other sides of the aisle, which is missing so much in traditional research, in the hobby side, which I hate the word hobby, because to me,
I hate the word hobby too.
This is my lifestyle. This is my life. It's not my livelihood. It doesn't pay my bills, but it is why I wake up every morning. Like I go to work because turtles exist. I have a beautiful family, but I do it all because turtles exist. And so to me, like a hobby is it's not what that
big
is
enough
to
of a
me.
word. Yeah.
It's not big enough of a word. Like it's not like cleaning nasty turtle water is not a hobby.
What you do
like
for
I would
fun.
not choose that as my hobby. This is my life's work because I think it's important. So anyway, I just think that's so cool. Like Zach, we should wrap up here. I just like I can't thank you enough. I can't tell you how much of a fan of yours I am.
Thank you.
Being able to hear your voice on our podcast makes me really happy because I know your voice so well now. And like, it's kind of like when you meet someone from the radio and you're like, oh my gosh, that's what you look like. That's cool. I listen to you between the podcast you're on as a guest and then now your podcast. Almost every week I'm listening to you. So it's awesome to have you on our show. I can't thank you enough for your time. And I can't thank you enough for everything that you're doing for the animals of the world and the students of the world and helping them, grow and change the world for the better. So this is just awesome.
That education piece is a big part of who we are. And so we're excited to see what you're doing.
Don't put yourself to sleep, Steve. Come on.
Excited to see what you're doing, hear about the work, and look forward to talking more in the future for sure.
Yeah, sure. I'll come back on anytime you want me.
I love it. Maybe we'll make it an annual thing. Just so
there you go.
I'll keep notes through the whole year for like questions. This is a question for Zach.
Yeah,
for sure. Yeah. Awesome. Thank you, and thanks everyone for tuning in. Episode 86, and we'll see you guys next time.
Thank you all.